Well, good evening, and welcome to BGSU's Science Cafe for this month. We have a wonderful lineup, and I'm lucky to have the job to just introduce and welcome. The Science Cafes are an initiative between several campus centers of excellence, which are really trying to invite people from our campus community and from the broad community to engage with some of the faculty and students and scientists and researchers of various disciplines on our campus to really think about the ways that science really impacts the communities we live in, work in, care about, and love. So we have a great lineup today. I am going to turn the floor over to my colleague, Kevin McClooney, who will really set the frame for this evening's conversations and presentations. And you'll have plenty of opportunities to ask questions at the end. So keep making notes, and I'll look forward to sharing questions and answers with you after our presentation. So again, welcome to BGSU Science Cafe.
Thank you, V. Welcome, everyone. So I'm Kevin McClooney. I'm one of the co-hosts of the Science Cafe. I'm an associate professor in the Department of Biological Sciences here at BGSU. I focus on aspects of global change in aquatic and terrestrial ecosystems, and I'm a member of the BGSU Center for Great Lakes and Watershed Studies. So that's enough about me. I'm very pleased to introduce you to Dr. Hans Wildschutte, an associate professor in the Department of Biological Sciences at BGSU, whose lab is right down the hall from me. Dr. Wildschutte received his bachelor's and doctorate from the University of Pittsburgh, followed by a postdoc at MIT, before joining the faculty at BGSU. Today, he will be sharing a bit about his very important research, discovering new ways to fight antibiotic-resistant pathogens. So please join me in welcoming Dr. Wildschutte.
Thank you, Kevin. Can everyone hear me? Can you see the slides? Great, okay. It's a pleasure to be here and talk about the research that my lab and colleagues are doing concerning new methods to inhibit deadly bacterial infections. My talk is going to be broken up into two aspects. The first part is going to be about just general information on antibiotics and pathogens. And the second part is going to sort of getting into the research on the methods that we're doing to help treat these bacterial infections. So we're going to talk about new antibiotics and phage therapy. And that work has generously been funded by the Cystic Fibrosis Foundation and the National Institutes of Health. So I'm just going to start right away and discuss what the main problem is. All right, so there are hundreds of thousands of deaths worldwide due to antimicrobial resistant infections, and that's what this figure shows. Right, so we know that there are many other events which cause terrible deaths. I mean, some of these are road accidents. Cancer is the leading cause of death. What I want you to focus on is the middle circle of this figure, where there's 700,000 deaths worldwide every year due to antimicrobial resistant infections. Right, so we really don't hear about these in the news. It's a rather large number. There are other events that are well publicized, and I just want to mention a couple of those that aren't in this figure. Right, so one of them is the opioid crisis, which we are facing. This is terrible in itself. In the US, there are about 70,000 deaths per year. Worldwide, there are about 250,000. Right, even though this is just as a terrible tragedy, these are about a third of the deaths that, in comparison to antimicrobial resistant infections. And another event, which I'm certain that you're aware of, is the current pandemic that we're facing. And unfortunately, we're reaching a milestone, which is about 5 million deaths worldwide in the past year and a half. If there's any light at the end of the tunnel with this pandemic, is that we have 3 vaccines that have been developed. Unfortunately, with antimicrobial resistant infections which are caused by bacteria. There are no vaccines to prevent these infections. So if we don't identify or discover new antibiotics and new treatments to treat these infections, right, this is going to lead to the leading cause of death worldwide within 30 years. So it's going to surpass cancer. It's going to be about 10 million deaths per year, right, which is twice what we've experienced with COVID. So this is a serious problem that we have on our hands. It's not really publicized a lot, and it's going to continue to get worse if we don't do anything about it. So that's the problem. Now I just want to sort of step back and talk about what are bacteria and antibiotics. So bacteria, right, these are small single-celled organisms. We can't see them with the naked eye. They're the most abundant living organism on Earth. They're found everywhere, and they're in the deepest parts of the ocean. They're in the Great Lakes. They're in soil. They're on us. They're in us. Most of them don't cause disease. On the contrary, they're actually pretty beneficial. They produce about half of the oxygen that we breathe. They break down food that we eat. They're involved in the carbon cycle, the nitrogen cycle. They also produce antibiotics, right? So they're very beneficial in a lot of aspects of our life, but there's a handful of them which cause disease, and those are what I call, or what I refer to as pathogens. So on the left-hand side of this figure, there's just a handful of these bacterial pathogens. One that you might be familiar with is Staphylococcus aureus. It's those pink round circles in the bottom left-hand corner of the image. Staphylococcus aureus, which is resistant to antibiotics, is called MRSA, methicillin-resistant Staphylococcus aureus, and it's a pretty serious skin infection. So if you are unfortunate to become infected with one of these bacterial pathogens, you can go to the doctor. If your immune system doesn't clear it, you can be prescribed antibiotics. Right, so what are antibiotics? As I mentioned, these are produced by bacteria, and we harness these, right? We collect these, we identify them, we turn them into medicine. Those are our antibiotics, which can cure the infections. One important thing about this is they're not effective against viruses. So if you have COVID, which is caused by a virus, and you go to the doctor and he or she prescribes an antibiotic, right, that is not going to do anything to cure COVID, which is a viral infection. So it's really important to know that these are used solely to treat bacterial infections. But the problem with antibiotics is we have overused them and misused them to the point where these pathogens are now resistant to antibiotics, right? Our antibiotics are failing, and that's the crisis that we're facing globally. So some of the reasons for the misuse and overuse of antibiotics are shown on this slide. So these are just three of the more important examples that I want to discuss. First one is overprescribing. So as I mentioned, if you go to the doctor, right, and they don't identify that you're sick from a bacterial infection and they just give you antibiotics, that's a misuse of antibiotics because they don't know if it's a bacterial infection. Secondly, if you are infected by a bacterial strain and you are prescribed antibiotics, you have to take that full course of antibiotics because it's important to kill that pathogen that's in you. And third, probably one of the most problematic overuses and misuses of antibiotics is them being applied in livestock and fish farming, right? So eventually, right, these antibiotics make their way to the environment, and that is the main cause which allows the evolution of these bacteria to become resistant to all antibiotics, and I'm going to explain how that happens next. So, right, if we're looking at circle number one, these are bacteria in the environment. The blue bacteria are susceptible to antibiotics. The red bacteria are resistant to antibiotics. And in the environment, you'll see, right, these red or orange bacteria are naturally resistant to an antibiotic, right? That's just occurs naturally. So if we look at circle two, when this environment is exposed to a particular antibiotic, all the susceptible bacteria die and the resistant ones remain and they multiply. So we see in circle three that they're thriving in the environment, and now when they're exposed to that antibiotic, they're resistant to it. What's even more problematic is they can spread these resistant mechanisms to other bacteria and pathogens, which we see in four. So now pathogens which were not resistant, right, can now they are resistant to antibiotics, which is a problem when people become infected. So if you are properly diagnosed and you need antibiotics, make sure that you take the full course when they're properly prescribed. right? If not, right, that's one of the reasons why we have these superbugs, right, which you probably are aware of that term, which is these pathogens which are resistant to most of the antibiotics, right? And they're terrifying and deadly. Right, so that reminds me of a joke. You can stop me if you've heard this one, but there's two people, right? They're walking into a Halloween party, right? The person on the left is dressed up in a ghost costume, and he's saying to Beatrice on the right-hand side, who was one of my previous students in my introduction to microbiology class, he's like, what are you supposed to be, right? I'm a ghost, I'm a classic Halloween costume. She's like, I'm an unfinished course of antibiotics. And she goes on to say, aren't I terrifying? Because I can lead to antibiotic resistance, right? So we know this really is a problem which is leading to antibiotic resistance, right? And it is indeed terrifying. And with Halloween around the corner, if you're looking for a costume, right, this is a great costume. Usually, in the past few years, I've been wearing this, and it really scares a lot of people, right? But in clinical settings, right, the problem is usually, well, you can go in, you can have surgery, you can come out, and as a result of that, if the technique is not sterile, you can acquire an infection. And this happened to a friend of ours just last year. She went in for a routine outpatient ankle surgery. Within a week or 2 weeks, her ankle looked like this, which you see on the left-hand side. A week after that, right, it looked like this, right? So I think, right, you don't have to be a doctor to see that this is a serious infection. This is MRSA, methicillin-resistant Staphylococcus aureus. They went down the list of antibiotics. It was the final last resort antibiotic, which was able to cure this infection along with 6 additional surgeries in 3 months. Right, so if it wasn't for that last resort antibiotic working, right, the only thing left to do is start amputating limbs. So they would have to, right, probably cut off her leg from the knee down, because if this infection makes its way to the bloodstream, it's deadly, right, because you can't treat it anymore. So sort of summarizing our whole sort of crisis, our whole problem is shown in this slide, and there's 2 aspects of this. One is that these pathogens are evolving multi-drug resistance, and number 2 is that new antibiotics are not being discovered. So I just want to go over this slide. On the x-axis, we see time in years, and on the y left axis, we see the percentage of pathogens that are antibiotic resistant. And those antibiotic resistant pathogens are shown by this yellow line and the 2 green lines. And this just isn't happening with these 3 pathogens, but it's happening with all pathogens. So we can see that, right, they are evolving multi-drug resistance. And concurrently, this blue line shows the number of new antibiotics that are being made available to the public for use. So we can see that these are declining. And in part, this is because large pharmaceutical companies no longer pursue antibiotic discovery because they don't profit from it. So you see these 2 blue flags at the top. In 1990, there were 18 large pharmaceutical companies involved in drug discovery. In 2011, there were 4 large pharmaceutical companies involved in drug discovery. Now there are 0. Right, so we have this problem, right, described by the perfect storm of the World Health Organization, that you have multi-drug resistant pathogens and we have no new antibiotics to treat them, right? So this is a serious problem, which is going to lead, right, to those 10 million deaths worldwide in 30 years if we don't do anything about it. Right, so, right, there's some things that you can do, which hopefully you are aware of, right? So this is sort of a flyer by the World Health Organization. 5 main points: use antibiotics only when they're needed, when they're prescribed by your doctor. Take the entire course. Don't save any. Don't give them to anyone else. And this last point, right, which has been stressed during the pandemic, right, make sure that you wash your hands, have good hygiene, avoid contact with sick people, and keep up to date on your vaccinations. Right, this just wasn't, right, discovered with the pandemic. We've known this for about 200 years. Right, so just in summary for this first part, we know that there are new methods that are needed to treat these deadly bacterial infections. Pathogens are evolving resistance to all known antibiotics. There are no new antibiotics at this time, right? And vaccinations, right? We can't develop vaccinations to treat these diseases, so we need to come up with some other treatments. And that's what I'm going to talk about next. Okay, so the first part you probably know about, trying to discover new antibiotics. I'm going to talk about that. And then the second point, phage therapy, is very interesting and compelling and seems to work really well. And I'll give a few examples of that to end the talk. But this is our antibiotic discovery approach. And since we know that antibiotics are produced by bacteria, we isolate bacteria from soil and water environments. We grow them in the lab. And then on the right-hand side of this upper figure is our antibiotic production assay. And I'll talk about how we do that. But the idea is if our environmental strains can kill multi-drug resistant pathogens, then that suggests those environmental strains are producing something new. So then once we identify environmental strains that can kill these multi-drug resistant pathogens, we use molecular techniques to identify the genes and then isolate and characterize the compound and see if it's something new that can funnel into clinical trials. Okay, so isolating bacteria. We have a collection of about 10,000 environmental strains which have been collected all over the world. Most of them are within the Midwest, United States. We do have some samples from Europe and India. But this is one of the first things that when a student enters my lab, I have them do is think of an environment which may be producing bacteria that are, or that may have bacteria that are producing novel antibiotics. So we have quite a lot actually from the Great Lakes as well, which we see in the bottom left-hand corner. This picture here, again, this isn't me coming up with these ideas, but these are my students. There's a body farm at the University of Tennessee in which they analyze dead and decaying bodies. So one of my undergraduate students wanted a soil sample from underneath a dead body to isolate bacteria. So we were able to do that. We isolated bacteria. And then once we have these, we take these samples to the lab and we put them on media to allow bacteria to grow, right? So we process these samples, grow the bacteria, and then those plates look like this. So after a couple of days, we can see that there are colonies of bacteria. They're different sizes or different shapes, indicating that they're different types of bacteria. And then we have to separate them from everything else. So we streak them out on a plate. You can see them growing. So this is all one type of bacteria. And then we take one of these colonies and we put it in a well. So this block contains media. You can see media, which is growth media, being dispensed in each one of these wells. And then we put a bacteria in one of these wells. There's 96 wells, so if you're looking down on this plate, this is what you see. Right, so each one of these wells has a different strain of environmental bacteria. And then we use these to test for antibiotic production. And that's done using this assay. So we take those strains in that 96 well block, and we transfer them using this pin replicator onto media which has the pathogen growing on it. So this brown indicates the growth or the spreading of an antibiotic resistant pathogen. And then these tan dots are the environmental bacteria. Right, so we transfer the environmental bacteria onto the pathogen, we allow them to co-grow, and then we screen for a zone of inhibition around the environmental strain, indicating that that environmental isolate is killing the multi-drug resistant pathogen. Right, so if we can identify this, right, that suggests that those environmental strains may be producing a new antibiotic, right? Because these are multi-drug resistant pathogens. These are pathogens that we have from a collaborator at the University of Michigan who is a clinician, and these are multi-drug resistant pathogens which came from sick patients. So I'm going to be telling you about a collection of 194 environmental bacteria that we isolated right just up the road from the Maumee River. And we're testing them against a panel of 28 multi-drug resistant pathogens. So all of those interacting with one another, we have about 5,432 individual interactions. Okay, so here are the results. So this is actually what the plate looks like. And we can see indicated by the black arrows, right, there's actually some of these environmental strains are able to kill the multi-drug resistant pathogens, right? So that's remarkable, right? These are deadly pathogens. So out of the 194 environmental strains, right, we were really excited and surprised about this, that 173 or 60% were able to inhibit these deadly pathogens. Of these, 36 can inhibit more than half of these deadly pathogens. So if you take a look, right, so these on the bottom here is the results of all the environmental strains and their ability to kill the pathogens. On the far right-hand side are the pathogens. Along the top are the environmental strains, and each one of these dots indicates that that environmental strain can kill a particular pathogen. So if we take a look at the far right-hand side, this strain, RC2C2, can kill 1, 2, 3, 4, 5, 6, 7 different pathogens. Okay, so we pick environmental strains which are able to kill, right, more than one of these environmental or more than one of these pathogens, and then we use different molecular techniques to try to identify the genes that are involved in producing the antibiotic. So I'm going to tell you about results with just one of these strains. We've done this with quite a few, and this is RC3H12, and it was able to kill or to inhibit the growth of 11 different multi-drug resistant pathogens. Right, so after we do some techniques, we identified that these genes, right, so these are the genes that are in the bacterial strain, are responsible for producing the antibiotic that is killing these multi-drug resistant pathogens. Right, and then after doing some bioinformatic analyses, we were able to determine that this is a rather rare and undiscovered gene cluster, and we could do other studies to predict what the compound looks like. Right, and we think that this is a lipopeptide antibiotic, and there are other antibiotics that are also lipopeptides, such as daptomycin, which have been used to treat bacterial infections. So we think that we've identified a strain which is producing a novel compound. Not only this strain, but we have a few other candidates which we are trying to isolate and characterize the compound. And that's something that our collaborators are very good at doing, and that's their lead in the project. So that's an overview of our antibiotic discovery approach. And this takes a lot of time and effort, a lot of people, and this is just sort of the workflow of antibiotic from the discovery to production, to FDA approval. Right, so we're involved in this first part, hit to lead and lead optimization and preclinical trials. So once we identify what we think is a new compound, of course we have to do testing in human cells and animal models, and if they pass that, then it can go into clinical trials. But as you can see, right, this takes a lot of time and a lot of money. Right, so that's what we are working on as far as new antibiotics go. So phage therapy, this is, instead of giving patients antibiotics, right, we give them phage to kill the pathogen. And some of you probably may not have heard of this, but bacteriophage or phage are viruses that specifically kill a particular pathogen. So here we see red phage that are attacking a pathogen and will kill it. So just like COVID infects our cells, these phage infect a particular pathogen. Right, so how do they kill a pathogen? Right, we see the phage binding to a pathogen, it injects its DNA, it makes more virus particles, and then it kills the pathogen because it breaks it open and release all those phage progeny. Right, so what we are doing is we've been able to isolate phage that can do this process with many multi-drug resistant pathogens. Right, so we can take these phage now and put them in clinical trials for people that have multi-drug resistant infections which are not responsive to antibiotics. And this on the right is just a picture of one of the phage that can infect a certain type of pathogen. But this phage therapy, right, so this whole process when getting phage and putting it into a patient, that's called phage therapy. And this just wasn't invented, but it was first used in 1919. But with the discovery of penicillin, the United States, for one reason or another, decided to pursue antibiotic discovery instead of phage therapy. Now, since we're at the crisis of multi-drug resistance with antibiotics, we are returning back to phage therapy. So I just want to mention two examples which have been successful and rather remarkable in treating patients which otherwise would have died. So here, let's see. So this is Isabel Halloway. At the time, she was 15 years old. She's a cystic fibrosis patient. She had a double lung transplant. After surgery, she became infected with a multi-drug resistant infection. And you can see it here, right? It's breaking through her skin. You can imagine what it's doing on the inside of her body. This is her mom on the right-hand side. This is Graham Hatfull, and this is the lead physician. So Graham Hatfull has a collection of about 15,000 virus or 15,000 phage, which possibly could infect this pathogen. So they actually reached out to him and they said, hey, can you screen your 15,000 phage to see if it can kill this pathogen, which is killing Isabel? And they were able to do that. And this, explanation by myself really doesn't do justice. So there's a little video here that I would like to show you.
This is Isabel. She was born in London and with cystic fibrosis. At age 15, she needed a double lung transplant. There were complications. She contracted an infection and her liver began to fail. She was given less than a 1% chance of survival. Because the infection in Isabel's body resisted every antibiotic her doctors gave her, Isabel's mom began searching for an alternate treatment, a new treatment, and what she found was Phage Therapy and the Hatfull Lab on the campus of the University of Pittsburgh. Phage Therapy matches an exact bacteriophage, a virus, with a specific bacterium to attack and kill it while ignoring the rest of the body's cells.
It was a hypothetical matter at that time because nobody has done that before. This would be a completely new and rather scary in some ways intervention to move to try to do that. On the other hand, we also appreciated that Isabel was out of options.
But the key to making this revolutionary and forward-thinking treatment work would be finding that exact match. And with Professor Graham Hatfull, who has dedicated his 30-year career to studying and collecting bacteriophages. He's become the conductor of a symphony of scientists, researchers, and technicians across 150 institutions with a model to inspire and empower more than 5,000 young diverse scientists from New Zealand to Nigeria. And in doing this, science for the sake of science, his team has amassed the largest collection of phages in the world. So with time running out for Isabel, Professor Hatfull and his team sorted through and experimented with the 15,000 phages in their collection to identify and genetically modify a cocktail of 3 phages to attack Isabel's exact infection. And as physicians in London administered the treatment, Professor Hatfull and his team, an ocean away, waited. Within weeks, Isabel's body started to heal, and while she is not fully cured, the infection is under control. Isabel is back in school, learning to drive, and living the life of a normal teenager.
Very few people in the world work on these particular types of phages. I'm not sure that there was anybody else in the world that could have done, and so that feels very special that this team of people was able to put that together.
This was the first time bacteriophages were used to treat a mycobacteria infection and the first use of engineered phages. Researchers hope that this is a transformative moment, one that could become life-altering and life-saving for the millions of people around the world who contract antibiotic-resistant infections each year. And it is why researchers, physicians, and patients across the globe are now reaching out to the University of Pittsburgh. Because of the work and vision of these pioneers, thinkers, disruptors, and champions, there is nowhere else on the planet where this could have happened.
So yeah, that's a rather remarkable story. And I have one more example, which just as surprising. So here we have Stephanie Strathy and her husband Tom Patterson. They're world travelers. They were vacationing in Egypt, and he started becoming sick. Again, this was a multi-drug resistant infection, different from the one that Isabel had, but still just as deadly and also resistant to all antibiotics, right? So he wasn't getting the proper treatment in India, or in Egypt. What can you really do? So they transported him to Germany. and then back to the United States. But still, even with the best, being in the best hospital on the West Coast, there still isn't anything that you can do, right, when you're being attacked by a multi-drug resistant pathogen. So on the right-hand side, right, he was in a coma, right, for about a month, undergoing multi-organ failure. Stephanie was able to reach out to scientists. They found phage, which were able to infect and kill the pathogen. They administered it to Tom, and within a week, he was waking up from his coma, and he is alive. The infection is cured, and it's a rather remarkable story, just like Isabel. She published a book on it. It's called The Perfect Predator, a scientist's race to save her husband from a deadly superbug. So I suggest reading that if you're interested in this. And I want to keep this in mind because this is a serious problem. You probably know someone that has been infected with multi-drug resistant pathogen. And if it is not responsive to antibiotics, I mean, who can you reach out to if antibiotics fail? Right? I know it's Halloween, but please don't think of calling the Ghostbusters. Instead, Stephanie Strathy started one of the first phage therapy centers in the United States. So it's called the Center for Innovative Phage Applications and Therapeutics. And she was one of our collaborators, and the phage that we find that are able to kill these multi-drug resistant pathogens, right? She's our gateway to clinical trials. So it's going from benchtop to clinical trials for patients that need it. So in summary, pathogens are evolving resistance to all antibiotics. Antibiotic-resistant infections are predicted to be the leading cause of death by 2050, right? 10 million deaths per year, twice of COVID and surpassing cancer, right? So vaccines won't fix the problem. Instead, we need new methods or other methods, new antibiotics, phage therapy to treat these infections. That's one of the things that my lab and collaborators do. And if you have to use antibiotics, please use them sparingly and properly. So, of course, right, there's a lot of people involved in this. This is just some of them here. These are some of the current and past students involved in drug discovery and phage therapy in my lab and in Ray's lab. So Ray is really spearheading this phage work. John LaPuma is our clinician, where we obtain our multi-drug resistant pathogens from. Kusan Ju is our biochemist expert, and he's involved in purifying and characterizing the compounds. And of course, Stephanie is our gateway to clinical trials. So that ends what I have to say. I have a slide here with just some ideas that we don't have to discuss now, but I mean things that you might just want to think about later on in the night or more in your life.
Thank you, Hans. That was a great presentation. Thank you. So I'm going to start the section where we do question and answer. I'll be the moderator for that. If you have questions for any of the panelists, which Dr. Wildschutte will introduce in a minute, please either place your question in the chat, which you can direct message to me, or raise your virtual hand in Zoom by clicking on the raise hand icon, and you'll have the potential to ask the question yourself. But first, Hans, can you introduce us to your panelists tonight?
Sure. I mean, we have two panelists. One, as I mentioned, Dr. Ray Larson, who's a close collaborator at Bgsu, and Liz Harper, who's a graduate student in my lab, who works on the phage therapy project as well. But I'll just let them sort of give a quick introduction of themselves. So I guess Ray can go first.
Okay, yeah. So hi, I'm Ray Larson, and it's really been a pleasure to be on this journey with Hans and with his students and with some of the students in my laboratory as well. We are busy isolating phage, not just we're looking at a fairly tight range of phage right now. We're focused mostly on organisms that are associated with cystic fibrosis, but at the same time, there are many other potential targets. not simply just those that involve human infections, but as Hans alluded to, part of the problem with antibiotic resistance comes from the heavy use of antibiotics in agriculture, not just farm animals, but also in aquatic fish farming and so forth. And so we've had a number of discussions where we hope to potentially branch some of our work into looking at some of those pathogens as well. So I think that certainly if you can control disease in animals that are being bred as food without using antibiotics, I think that makes it a very positive contribution. So I think that's one of the places where we're looking. And other than that, I think I'll pass it off to Liz. She can give you a graduate student aspect of these projects.
Everyone, my name is Liz, and I'm a second year master's student in Dr. Wildschutte's lab. So I joined his lab last August, and I've been working on my project for just more than a year. My undergraduate degree was in chemistry and biochemistry, so then I made the switch to microbiology, and it's been really exciting to learn about phage and the project that I'm working on. I'm using the phage as a selective pressure to find phage that can evolve a trade-off in Pseudomonas aeruginosa between phage resistance and antibiotic resistance. And now I'm getting to the point in the project where I get to work with the molecular techniques where I'm looking at the genomes and the mutations in them. So that's been really exciting.
Great, thank you. We've got some questions starting to come in, but I'm gonna start with my classic question that I like to ask every time. And this is for each panelist to answer. How did you get interested in science? What do you love about it? You can start with Dr. Wildschutte.
Okay. I mean, I've always loved science. I think it just started with the outdoors, but then, really, when I was an undergraduate, that really sparked my attention. I mean, it was actually after I obtained my degree at the University of Pittsburgh. Actually, I was quite familiar with Graham Hatfull, who did the phage therapy work. And I took my first microbiology class after I graduated with my undergraduate degree. And then from then on, I knew I wanted to pursue a career in microbiology because it was just so fascinating that these creatures that are so small are just involved in every aspect of our life. So it just sort of took off from there. Liz?
Well, I kind of had taken a windy sort of path to science. As a kid, I was sort of nerdy, and then I had a favorite atom and I had a favorite planet and stuff, but then I kind of wound my way around from that. And then as an adult, I found my way to massage therapy, and I started learning about, the body, and there are all of these tiny processes that our human bodies share with all of the other living cells. And I think that that's what really started to fascinate me, was that connection in the cells and the cellular processes like cellular respiration and things like that. And so I started to get more and more interested in that. And I was working as a massage therapist, and then I got the opportunity to go back to school and start studying science, and just became more and more fascinated with microorganisms and biochemistry and all of those processes that we all share.
So I got my interest in science probably when I was 5 or 6 years old, and I had a babysitter who had the Time Life Science book series, and I would spend every afternoon at her house looking through the pictures and reading about the things, and it was all really fascinating. And so then, having discovered science, I think anything that had an ology at the end of it, at one point in my life, I was going to be that kind of ologist. But it wasn't really until I was in high school and had we had a microbiology class in high school, which was kind of unusual for most schools, but I found it really interesting. And in particular, what I found interesting was coming to learn how much of an impact infectious diseases has had on the development of culture and how it has shaped the way the world is today. And so the combination of the science components and the heavy influence it's had on the development of cultures worldwide is really what has kind of kept me interested in this. And also it's nice to have that big playpen called a laboratory to just spend your day in.
Thanks, everyone. Great answers. Okay, so I want to go to the chat, and we've got a question here. So if we can continue to find these natural or environmental bacteria and use them to inhibit potentially deadly viruses, do you think that would allow us to eventually prevent certain types of infection from occurring in the first place?
So I guess is the question, should we take antibiotics before we get sick? Or I don't.
Potentially. I mean, I'm not sure, but I think they also were suggesting that is there a way to use these technologies to prevent the infections from occurring?
So we do have. Antibiotics are sort of the last resort. We have an immune system which really does a remarkable job of killing pathogens which we encounter every day. So taking antibiotics before you get sick is actually a misuse of antibiotics. So you only want to use them when you absolutely have to. And again, right, they are based on the mechanism on how they kill. The only things that they work for are bacterial infections. So viruses like COVID, the flu, antibiotics do nothing for that.
That's an important clarification. Thank you.
I would add another aspect to that, though, in that, of course, outside of the bacteria that we worry about infecting us, we have our own natural bacterial flora. Every surface of our body is covered with bacteria. The vast majority of cells that we have aren't our cells. They're the microorganisms that live on us and within our respiratory tract and within our digestive tract and so forth. And they play a very strong role in protecting us from many infectious organisms simply by being able to outcompete many pathogens for their preventing them from being able to colonize our surfaces and then gain greater access. So I think something that we can do proactively is to maintain a healthy body flora, which we're only now starting to identify ways to do that. But certainly it turns out that our diet is very important, and there are other aspects to that too. So while certainly we don't want to be taking antibiotics that would simply accelerate the opportunities for bacteria to develop resistance, we certainly should encourage and foster having a healthy flora, so that we can exploit what that provides for us by good nutrition, by good health, by exercise, all of those things that I think go to maintaining a robust physiology and not only nurture our own flora, but also our immune systems. I think that we can certainly be proactive about reducing the possibility of getting those kinds of infections.
And one other thing to add, that's a good point, Ray, but antibiotics just don't kill that pathogen that's causing a disease, right? They kill a lot of the bacteria, the good bacteria that's in your body. So you just don't want to take antibiotics, right, if you don't have to. That's one of the benefits of phage therapy, is those viruses are specific and they just target that one pathogen. They don't target your cells, they don't target any other bacteria. So yeah, I mean, you don't want to take antibiotics unless they're absolutely necessary.
A very important point. So I've got another great question here in the chat. Is there a Great Lake that is best to gather bacteria samples from? And I'm going to add, because I had also sort of written my own question that's highly related, and make it a little bit broader. Are there certain places from which you or your students have collected that have been more productive in terms of finding new antibiotics or phages? And make sure you answer the question about the Great Lakes too.
Yeah, so Yeah, we're not sure. That's one of the things that we're trying to determine is what's a good environment that might enrich for these antibiotic-producing bacteria. We've isolated quite a few samples from the Great Lakes, and it does, comparing them to bacteria that we isolate from soil environments, it seems like water-derived ones are more effective at killing pathogens, and I'm not exactly sure why that is. But I specifically mentioned the results from the Maumee River because those seem to be the most active ones that we've seen. I mean, all of that killing is rather remarkable. And sort of the idea that we're coming up with and we have to test is polluted environments, right? Ones where there's a lot of nutrients and a lot of waste. You can imagine that's a source of food for bacteria. So bacteria produce antibiotics to outcompete other bacteria for this food. So we're hypothesizing that these sort of nutrient overload environments are good habitats that enrich for these antibiotic producing bacteria. So one thing that we plan on doing is isolating some bacteria from Lake Superior, which is considered a clean environment, and comparing those to other polluted water environments, right, to test our hypothesis that sort of these nutrient environments select for antibiotic producing strains. So I think that addressed both of those.
That's great, thank you. Okay, so this is maybe a very simple question, but I think it's a great one to sort of clarify the collaborations a little. Does Bgsu send phage samples off to the University of Pittsburgh to become part of the collection of phage diversity?
Oh, this is an awesome story in itself. So when I was a graduate student at the University of Pittsburgh, Graham Hatfill was actually in the lab right next to me, and he started this program now known as Phage. But at the time, it was just with high school students, and they would go out and they would collect samples, and they would isolate these phage, which can infect a particular strain. And that's how he grew this collection of 15,000 phage, right? So when Isabel came sick with this pathogen, which is sort of related to the one that Graham works with, right? All of these 15,000 phage that he has in his collection were essentially collected from high school students and undergraduates. So it's really, right, I know Graham personally, and we're more of a personal relationship as opposed to a collaborative relationship because, the phage that he works with is very different than the phage and the pathogens that we work with. But yeah, I mean, I'm in contact with him constantly, just bouncing ideas of phage therapy and other, ideas.
Great, thank you. Let's see, let's go to...
Actually, Kevin, real quick, this is Ray. I would simply add to that to say that we've been running the Phage program here for about 4 years now, and we kind of have truncated it during the time of COVID, but we hope to come back strong with it. But at this point, we've contributed somewhere between 50 to 75 phage to that collection that's at Pittsburgh, and like many other universities that have a phage program, we're all feeding into that.
Great. This is a really interesting, thought-provoking one. Is it possible to treat non-bacterial pathogens with viruses?
That's interesting.
I could
Go ahead, Liz.
chime in. I would say I don't know for sure because I'm inexperienced at that, but my hypothesis would be yes, because there seem to be viruses that infect basically every kind of cell on the planet. So viruses infect fungi and insects, all different kinds of animals and plants. So it seems like if perhaps we had a yeast infection, so a pathogen that came from the yeast family, we could find viruses that infected the yeast and theoretically we could develop a phage therapy that worked that way. But I'm not aware of any specific research in that direction.
Yes, and I'm not aware of any clinical trials as well. Actually, well, there was a documentary on using an adenovirus to attack cancer cells, and I don't know. I think there was quite a few patients that died in that clinical trial, so I think they halted that research. But yeah, I mean, with phage therapy, with attacking bacterial viruses, it seems very promising. And I'm not well. I don't know of any other therapies to use to treat non-bacterial pathogens.
Yeah, I would go along with what Liz said, that at this point it's mostly hypothetical, but as we have only in the last couple of decades come to be aware of the importance of our bacterial flora, we're now coming to recognize that humans have a normal viral flora that while we always think of viruses as being absolute parasites and pathogens, in fact, there are some viruses that seem to be very well co-adapted to us. And so the possibilities that they are also potentially contributing to our well-being and could potentially be enhanced in that is something that I know some people are starting to speculate about. And hopefully there'll be a future there where it'd be nice to have a virus that gets rid of tapeworms. That would be a whole lot better than I think some of the other approaches people take to things like that.
Great, thank you. Yeah, it's really cutting edge ideas. That's really cool. So we've got a couple of questions in here, just wanting to know a little bit more about, I guess, where phage are found, how ubiquitous are these phage in the environment, and I've got a related question here that so the phage that was helping the young lady in the video, is that present all over the world? And kind of a related question of, let me go down to it, where phages are found and their life cycle.
I would say phage are found everywhere. Any place you have a bacterium, you will find a phage. And just to provide an example, once a week my laboratory gets sewage samples from 6 locations, from 6 sewage treatment plants.
Oh, you just muted yourself, Ray.
Every week my lab is getting sewage samples from 6 sewage treatment plants in Northwest Ohio, and every week we're screening those for the phage for the pathogens that we work with, but also for phage that are specific for a common human bacterium, E. coli. And we're getting anywhere from 10 to 15 new E. coli phage every week simply from those sewage samples. And so they're everywhere and relatively straightforward to find usually if we have the right hosts and the right culture conditions. Unfortunately, some of the ones we're really looking for seem to not pop up as quickly. The one particular phage that we spent 6 months looking for, and we ultimately found our first example of it in some deer droppings that I picked up in the plastic bag when I was in Pennsylvania. And so I walk around with plastic bags all the time and find things that most people wouldn't put into their pockets to bring back to the lab to see if we can find phage in them. And sometimes it works out wonderfully, and sometimes the laundry is kind of a mess. So
this is why people look at biologists weird. We're always collecting weird things. I've got a great question that I know that Hans will love to answer because I know I've seen him give a talk about it before. Mike, the question is there any concern that increasing phage would eventually induce the evolution of phage-resistant bacteria? And would this be as much of a concern as the current antibiotic resistance problem?
Yeah, that's a great question. And actually, I'll let Liz answer that because that's sort of what her project is that she's working on.
So the beginning part of my answer is that bacteria definitely evolve resistance to phage. but it's also part of a normal process of what we call like a host predator arms race. So it's hypothesized that the bacteria and the phage have been in this trade-off relationship where the phage infect the bacteria and the bacteria become resistant to the phage, but then some of the phage evolve changes that make them still able to infect the bacteria, and that process has been perhaps going on for billions of years. And so I don't know the exact answer about whether the phage resistance problem will get out of control the way the antibiotic resistance problem has. But my hypothesis about it is that because it's a natural process of going back and forth, that it won't be as rapidly a developing problem as the antibiotic resistance, because the antibiotics are chemicals and it's just a one direction evolution and there's no trade-off or arms race that goes on there.
Now, I'm going to just add a related question from another participant. Asked, is there a chance that these engineered phage may mutate to attack beneficial bacteria?
I guess can you hear me? I don't know if, yeah, okay, so, so the interaction between a phage and a bacterium is very specific. So, usually can kill and infect, right, only one particular strain. So the likelihood of it going and infecting other bacteria in your body is very low. But that's what antibiotics do. So I don't think that is an issue that will arise with phage therapy. One of the things that is probably a little bit more problematic is, you're injecting these things into your body and they're foreign. So sometimes there's an immune response to the phage, but we're using this right now as a last-ditch effort for people that are otherwise going to die. So in the case with Tom Patterson, he developed an immune response to the phage that they injected in him, but that passed, and the phage therapy ended up working. But they think he almost died. He was dying anyways, but they thought he almost died when they first injected the phage, just because it was a large amount of phage. It was something foreign to the body, and there was immune response, and he almost died from septic shock. But that eventually that didn't happen. Right, so it's a long way around that. These viruses are very specific, and they just target that particular pathogen, and them mutating to infect other bacteria is very low.
Thank you. Okay, we've got another question that looks like it could probably be like the subject of an entire class you might teach, but maybe you can just answer very briefly. How do you go from finding the bacterial colony that inhibits bacterial growth to determining the gene sequence for the protein that causes this to happen?
So there's a lot of molecular tools that we use to identify the genes. One of them is called transposon mutagenesis, where we randomly mutate the genome, and that particular environmental strain, which can kill the pathogen, when we randomly mutate the genome, we look for mutants that can no longer kill that pathogen, and that suggests that there's a mutation in the genes that are producing the antibiotic, and we use other molecular techniques to go in and identify the genes. And once we have once we've identified the genes, then we can get an idea of what the product is, and we use other bioinformatic techniques to identify if those genes have been discovered before and if that compound is new or not.
Great. 2 more questions, and then we'll wrap things up. When transporting bacteria from far-flung places, like India, for example, what must be done to keep them viable for use in science? Anything special?
So one thing about bacteria is they're very hardy. They can survive a lot of different environments. So what we do is we have filters, And if we're taking water samples, we pass the water through the filter, and the bacteria gets trapped on that filter, right? So they essentially stay dormant on that filter. So we just take that filter, right? We put it back in the bag that it came in, and we bring it home with us. We put that filter on media, and they're still viable and they still grow. So that's one easy way, right? Instead of transporting water or soil, right, across the country or across the world, we simply pass them through the filters and take the filters with us.
Cool. So I've got the last question I saved is to finish on kind of a fun note. This person says, I am a first-year student here at BGSU, and phage therapy has been a major interest of mine for many years now. Are there any opportunities for me to work in the lab?
Always opportunities to work in the lab, right, Ray?
Always. I think that the best person to get into your lab is someone who's interested and eager, and they've already got all the tools in place for success. So certainly, if anyone who's interested in pursuing this, we're happy to talk to you. We're happy to find potential circumstances either with us or with other people where you have opportunity to explore your interests. I think it's the reason you're here is to explore and learn. So please reach out and we'll let you. We're happy to work with you.
Wonderful. Okay, I'm going to turn it over to V, who's going to do kind of a little brief wrap up and maybe tell you a little bit about some upcoming science cafes.
Well, as ever, I'm at a science cafe where I've been entranced during the whole process. And the bit at the beginning was quite terrifying. But it's nice to end up with for those of us who have deep belief in how science both impacts our lives and our region. I'm leaving optimistic. And as we would say in England, dead impressed by the work that, again, our scientists are doing on campus. And I really appreciated having that sense of that connection across countries and different researchers, and appreciated the opportunity to hear from a younger researcher in the lab. So I really appreciate that modeling of the ways in which science is a very collaborative profession, and we're building on knowledge. So great thanks. And thank you to participants. I think we had a larger crowd than we'd anticipated, as some people must have joined us close to the start. And I appreciate those of you who've stuck through the questions at the end. And thank you, presenters. It's it's hard to go and talk to our peers at conferences, but sometimes I appreciate it can be hard in this online environment where you can't see all of those people who are sitting listening. But I leave again with a whole set of questions, and kind of wish that I'd taken a science track earlier in my career instead of political science and community engagement. But and we do have 2 more great science cafes coming up. And if you signed up today, you'll be added to our list if you're not already there to get an invite to our next 2 sessions. Kevin, I don't know whether you want to do a quick summary of what our next 2 sessions are.
I don't have the summaries pulled up right now. I wasn't.
I don't either.
Yeah.
Well, we'll make sure we have on board Patrick Caniglia, who is an undergraduate student in our office who's been doing a lot of the outreach and coordination. So we'll charge Patrick to both share an evaluation with everyone who attended, but also share the upcoming events. So again, thank you to all of our presenters this evening. I'm not sure whether I want to come and visit your labs, given what might, as I'm learning what might be there. But I really appreciate you taking the time, and I think I leave, to me, the measure is I always leave having learned something and having a deeper appreciation for the kind of scientific work happening, not just on our campus, but in collaboration, which I think your presentations this evening really demonstrated. So thank you.
Thank you.
Yeah, and thank you,
Yeah.
V, and thank you, Patrick. We couldn't do this without you. And I will mention that the very next one, will just briefly say, it's by a professor in the School of Art, and it's going to be really interesting about science communication. And so I encourage you to attend. I think it's going to be fascinating. So thanks everyone for coming.
And our thanks to our partner WBGU. I think their support has made this possible for those of us who believe in public broadcasting. And they have been a wonderful supporter, and I think demonstrate the commitment of public broadcasting to really bring great ideas and learning opportunities to a broader community. So thank you, WBGU colleagues.
Thanks, everyone. Have a good evening.
Thanks, everyone.
Thank you.